spiegare un attimo che cos'è stasera. Sono molto contenta di vedere tanti ragazzi e tante persone così scientificamente importantissime. Allora, stasera noi abbiamo pensato di fare questo meeting per il gruppo NAC, cioè Nana Alumni Community, e vorremmo farlo a cadenze regolari per invitare di volta in volta un ricercatore, uno scienziato che riteniamo possa essere di interesse per i nostri alunni. Non si sente bene? Ah, la musica forse? Sì. Pietro, scusa, puoi togliere la musica? Ah, già fatto. E quindi questa volta, perché noi sapete facciamo sempre il meeting più importante di Nana la prima settimana di dicembre, questa volta abbiamo invitato a parlare il professor Claudio Tripodo, che viene dall'IFOM di Milano e ci ha fatto questo regalo di venire e addirittura restare per tutta la durata del convegno, che è una cosa molto preziosa. Per spiegarvi naturalmente il valore scientifico del professore passo subito la parola a Stefano Casola, che è il coordinatore del gruppo di alunni.
Sì, quindi buonasera a tutti. Innanzitutto è un piacere di tornare qui e vedervi e trovarvi tutti quanti. Sì, è stato un anno in cui abbiamo discusso spesso con Donatella quale potesse essere il modo migliore per ricordare Francesca. E di nuovo, diciamo, la scienza è quello che possiamo fare, cercare in qualche modo anche di rendere questo momento scientificamente qualificato. E quindi abbiamo pensato di invitare o cominciare a una serie di keynote lectures che verranno tutte fatte rigorosamente in inglese, quindi mi scuso se qualcuno qui non conosce la lingua, ma la lingua della ricerca è l'inglese, quindi abbiamo deciso di rimanere tenendo questo livello e quindi nel ragionare sulla prima vera keynote lecture, che tra l'altro dedicata a Dato Boncinelli, ho avuto, voglio dire, il piacere già da qualche anno di conoscere Claudio, che per me è Claudio, e diciamo, al di là dell'amicizia, scusate se adesso mi emoziono, è chiaramente un ricercatore di primissimo livello, è una persona che, lo capirete dal modo con cui presenta i dati, è fondamentalmente spinto dalla curiosità che tutti i ricercatori devono avere, ma portandola a un livello di dettaglio che solo la morfologia, guardando una sezione, ti permette poi di comprendere. Quindi giusto per ricordare un po' la carriera di Claudio, Claudio si laurea in medicina a Palermo all'inizio del 2000, dopodiché decide di fare una serie di piccoli postdoc, se vogliamo, in giro per l'Italia, quindi va prima dal professor tedesco a Udine a studiare il complemento, per poi spostarsi a a Trieste, poi è andato a Udine da Carlo Pucillo a studiare mast cells, quindi è rimasto nel contesto dell'ematologia dall'inizio, pur essendo un patologo che copriva l'intera gamma, chiaramente, delle patologie dei vari organi e tessuti. Dopodiché ha preso una specialità in anatomia patologica, ha fatto partire il suo laboratorio dopo aver fatto, un altro importante passaggio nella sua carriera nel gruppo di Mario Colombo all'Istituto Nazionale dei Tumori, dove ha studiato ancora l'importanza della matrice extracellulare nelle sindromi mieloproliferative, e da lì ha deciso di tornare nella sua città natale, dove ha aperto un gruppo di immunologia dei tumori. Da quell'anno è rimasto fino adesso sempre finanziato dall'AIRC, quindi a dimostrare anche la qualità della sua ricerca, e fondamentalmente ha un curriculum dal punto di vista di pubblicazione e di contributi scientifici lunghissimo. Voglio solo ricordare che lui è stato il primo vincitore del premio Beppe della Porta nel 2018-19, fornito, tra l'altro conferito direttamente al presidente Mattarella, ha vinto una serie di altri premi in giro per l'Italia e l'Europa, è socio fondatore della Fondazione Italiana Linfomi, fa parte del working group di ematopatologi che copre, diciamo, soprattutto la componente, diciamo, di malattie che hanno origine dal midollo osseo, e quindi, diciamo, è una persona che poi ho cominciato a conoscere interagendoci a livello scientifico, è arrivato fino in IFOM qualche anno fa, da quel momento in poi non ci siamo più staccati, le nostre collaborazioni sono continue, il modo di vedere la scienza che ha Claudio è unico e lo verificherete nella sua presentazione. Quindi adesso veramente non mi voglio più dilungare, lo ringrazio innanzitutto perché è stato, voglio dire, un periodo piuttosto intenso per lui e nonostante tutto ha deciso di venire e condividere la sua scienza, quindi ti ringrazio personalmente a nome di tutta la fondazione.
Thank you. Grazie Stefano, thank you so much. It's a great privilege to be here with you on this occasion. I think that we can just start with this short journey through an unusual perspective on hematological malignancies, which is conferred by the study of the stroma, which is sort of a scaffolding, and particularly methodologies based on spatial resolved approaches. You know that pathologists are very familiar with the analysis of tissues, of cells and their relationships, including their spatial relationships. And now this spatial information is becoming more and more crucial for the identification of new clues, insights into these diseases. So going to the primary hematopoietic organ, the bone marrow, we can see that there is strict compartmentalization in this tissue. So you can easily identify an area in which precursor cells should reside near the endosteal edge of the bone trabeculae within our trabecular bone, and compartments that harbor differentiated cells. So the cells that should be ready to go into the peripheral circulation. And in order to understand fully how the spatial localization and zonation of these tissues is impacting on the definition of the alterations that become evident in several diseases of hematopoiesis, now we can exploit new methods that are actually single cell resolved, spatially resolved, and that also integrate the new frontiers of image analysis, pathological analysis, and AI, artificial intelligence, actually. This is the outline of a really interesting study from Cornell University in which they started profiling a series of normal bone marrow biopsies and myelodysplastic syndrome biopsies, and also biopsies of patients with clonal hematopoiesis of undetermined significance, which is a sort of pre-neoplastic condition that can develop with age in some individuals. And they integrated data from image analysis, from immunophenotyping, hyperplex immunophenotyping, with also AI technologies. And based on the integration of these methods, they could actually identify some features and quantify some features that defined pathological states. And those features are not surprising, are those that we already knew are needed to diagnose a pathological state like myelodysplastic syndrome or acute myeloid leukemia. But nevertheless, so far, all these parameters were just qualitative, more or less, with some quantification attempt made by pathologists on bone marrow biopsies. But using these very detailed integration of methods, they could actually define that you can measure the exact distance that precursors should have from a vessel in the intertrabecular spaces in order to be a physiological condition. And when these distances are either increased or in some circumstances decreased, this can configure some states of alteration in normal methylation of hematological cells, of hematopoietic cells. And this also applies to the quantification of the size and shape of cells. For example, megakaryocytes that are a lineage from which platelets derive can be measured, and their exact size or shape can be defined, and according to these, we can even try to subclassify cases as dysplastic or as cases with this megakaryopoiesis more or less pronounced. And this also may apply to a condition which is called ALIP, so abnormally located immature precursors, which was a condition that defined a state usually associated with myelodysplastic syndromes that pathologists normally identified by seeing clusters of immature CD34 positive, CD117 positive cells around vessels. But now we have precise topographical criteria to define the presence, quantity of ALIPs, and also we can try to stratify the cases according to the frequency of these abnormally located immature precursors and obtain a risk stratification of patients based on these quantitative methods. So basically, we are moving towards quantitative single-cell spatially resolved type of analysis. And the stroma has a role in all these alterations that we can now try to quantify. And it's not actually something new, because we already knew that there are specific stromal defects in the hematopoietic niche that can directly drive hematopoietic malignancies. So hematopoietic malignancies are actually related with the progression of a malignant clone through genetic events, but they can be triggered by a faulty supportive hematopoietic niche. For example, in 2010, David Scudden's group described the alteration of Dicer or SVDS genes in osteoprogenitors, which are the progenitors of the bone developing cells in the trabecular bone, as events that could drive the development of myelodysplastic syndromes or overt leukemia progression. So basically, a defect in a stromal cells, in a mesenchymal cells, deputed to synthesize bone matrix and actually to nurture and organize the endosteal niche, may unleash myeloproliferation, or in this case, myelodysplastic-like or AML-like states. So there is a very strict bond between the stroma and the malignant clones. And this is particularly evident for myeloid clones, but not only. You know, we have basically two hotspots of tumorigenesis in the hematopoietic system. The bone marrow niche, for sure, where most of myelodysplastic or leukemic or myeloproliferative diseases arise. And we have a second hotspot, which is actually the first in terms of prevalence and frequency, which is the germinal center of secondary or tertiary lymphoid structures in lymph nodes, for example, or other organs that harbor germinal centers. And the germinal center is another hotspot of tumorigenesis for hematopoietic cells. And again, it is a highly strictly compartmentalized environment. When you can identify a dark zone that is prevalently deputed to the proliferation expansion of B-cell clones that are going to undergo germinal center reaction for the affinity maturation of their immunoglobulin repertoire, and the light zone, which is mostly a region in which interactions with other cell types occur between B cells and T cells or follicular dendritic cells, and where the B cell receptor is challenged and matched for its ability to recognize cognate antigens. So within this very peculiar compartmentalized environment, you have specialized stromal core, and we are going to discuss about that. But again, we can study these complex environments by using new techniques that enable to gain a very detailed overview of the cells inside this microenvironment, maintaining the spatial resolution. For example, we can apply new methods of spatial transcriptomics analysis at single cell level as well. So we can probe the transcriptome of each cell within a specific setting and environment, and in this way we can try to reconstruct the interaction between B cells within the different contexts of the germinal center, interaction with T cells, and with these spatially resolved approaches, we can even try to define new markers, new diagnostic markers that can be useful for pathologists, for hematologists, and can be applied in routine diagnostics. Because most of these methods I mentioned are just for research. This is an example of a marker coming from our experiments on the germinal center spatial profiling, in particular the germinal center spatial profiling of T cells, and we obtain a transcriptome of the T cells that reside within the germinal center and compare with the transcriptome of T cells outside the germinal center in secondary lymphoid tissues. And among the markers that were upregulated in the germinal center is this histidine ammonial lyase, HAL, that, as you can see, marks the cells, the T cells that are scattered within the germinal center. Why is this important? Would it be important? Yeah, we still need new markers to define specific subset. And TFH is follicular helper T cells are the cells that help the B cells throughout the germinal center reaction and the production of the antibodies in the germinal center. And these TFH cells actually lack very specific markers. And today we need markers for the diagnostic of lymphomas that that originate from TFH cells. And this marker, for example, may be used to highlight some peripheral T-cell lymphomas with a TFH phenotype to complement other markers in identifying these subsets. So through spatially resolved profiling, we can derive also new markers that might be useful for diagnostic purposes. But the homology between the two hotspots I mentioned, so the bone marrow hematopoietic niche and the germinal center, is not confined to the fact that these are two hotspots of tumorigenesis in the hematopoietic system. It's interesting to note that in terms of stromal composition, environmental and extracellular matrix composition, the two environments are very similar. So the bone marrow endosteal niche and the germinal center are very similar in terms of stromal composition, even if we study the expression of several markers of specific mesenchymal elements or components of the extracellular matrix or some key chemokines that are expressed by specific stromal cell types, we see that actually the endosteal niche and the germinal center share a lot of these markers, but they might be contributed by different cell types. Let's have a look to collagen type 1. Collagen type 1 is a bone matrix collagen. So basically it's very abundant in soft tissues, but in particular in bone, and the endosteal niche is very rich in collagen 1, which is synthesized by osteoblasts, and then endosteal fibroblasts. But in the germinal center, it's mostly produced by macrophages. At the end, we still have abundance of collagen type 1 in the two environments. And how can we link this, this common stromal composition of the two environments? Is there any pathological link between the two environments? And the answer is actually there are some clues that the two environments may be associated in some way. for example, lymphomas originating in the germinal center, and particularly specific B-cell lymphomas and T-cell lymphomas of germinal center origin, like follicular lymphoma, when they metastasize to the bone marrow, where they colonize the bone marrow, they reside in the endothelial niche. So they go to the same niche that we saw is very similar in terms of environmental stromal composition. And so this is a prototypical paratrabecular pattern of infiltration of the malignant CD20 positive cells that surround the bone trabecula in this case of follicular lymphoma. And this extends also to other germinal center-derived lymphomas, while other forms of lymphomas tend to localize in regions that are distant from this endothelial compartment in the intertrabecular regions. And they tend to form different types of aggregates, like, for example, nodules, interstitial infiltrates, or intrasinusoidal aggregates within the vessels of the bone marrow, the sinusoidal network of the bone marrow. But even in this case, stroma becomes an element of heterogeneity. For example, some of these tumors that localize in regions that are not the hematopoietic niche in these intertrabecular regions, in the interstitial compartment, might integrate more or less within the pre-existing stroma. In the upper case, we see a splenic marginal zone lymphoma case that integrates perfectly within the CD10 stromal cell, follicular reticular stromal cell meshwork of the bone marrow. In the lower part, we see a same histotype of lymphoma, different case, in which the stroma of the bone marrow is, effaced. It's not actually present within the infiltrates, and the infiltrates are not integrating, intermingling with the pre-existing stroma. What is going on here? The lymphoma is modifying the environment in this case. We have a different kind of stroma inside the lymphomatous aggregate. And this different type of stroma can be identified as cells with a phenotype that is very similar to the core, the stromal core of germinal center, which is composed by follicular dendritic cells. Indeed, heterogeneity in the stromal reaction, in the stromatogenesis associated with malignancies of the hematopoietic system is a fact. And this can be extended also to experimental simplified models. Let's say, for example, to select a cell line and to inject the cell line into mice and then analyze the composition of the infiltrates in different organs. As we know, unfortunately, all these tumors are widespread, often in the organism. Most of the non-Hodgkin's lymphoma are already on an advanced stage when they are diagnosed, and also other hematopoietic neoplasms as well. And we have studied the infiltration of the bone marrow, the liver, and the spleen by a A20 diffuse large B-cell lymphoma model, and the composition of the stromal reaction. So the infiltrates of the lymphoma are populated by different types of mesenchymal elements, of stromal elements. And the composition is slightly different from site to site, from bone marrow to liver to spleen. So there is a different type of stromal reaction that is probably influenced by the nature of the pre-existing stroma. So the stroma, in this case, becomes an element of diversification of a same cell line in the different sites. And this also associates with the capability of the cell line to express more or less some relevant programs like MYC-related transcriptional programs or other programs that might be relevant for the lymphoma fitness and progression or for the integration within the microenvironment. And this is part of a phenomenon that is probably just selection. So when we think about different disease localizations, we should also think about selection that is differently ongoing at different cell sites and different infiltration sites. And this can be influenced by the type of stromal reaction that is associated. In this other experiment, cells of the same cell line that have been colored with different fluorophores are collected from infiltrates in the bone marrow, the spleen, the lymph node, or non-lymphoid sites like the liver. And the diversification of the clones, so the variety of subclones that are present in the infiltrates is different in different sites. In particular, non-lymphoid sites tend to select just more than the lymphoid or the primary hematopoietic site. So there is a different selective pressure that is also a function of the type of stromal reaction. In normal conditions, also the stroma is quite strictly defined in terms of zonation or spatial organization. For example, we have the follicular dendritic cells that very well mark the germinal centers or the follicles, and the smooth muscle actin, which is a marker of several fibroblastic subsets that is much more represented in interfollicular regions. So this is a very clear picture of what is going on in terms of stromal composition in a normal secondary lymphoid tissue, but this is not the case for tissues that are effaced and disrupted by a lymphomatous proliferation, where the heterogeneity might be very, very high. So we might have some cases in which we have still some compartmentalization of different types of stromal reactions and GFR, so an FDC-like stroma on the upper left in the first picture, and the smooth muscle actin-rich stroma on the lower part, and in other foci we have more interweaved or still segregated but less dense types of stromal reaction. And this is what we observe normally. So normally, ranging from a case to another, from a region to another, from a single localization to a different one, we might have different types of associated stromal reactions. And again, adopting spatial profiling methods, so methods that enable us to probe the transcriptional identity or proteic identity or genetic state of defined regions according to morphology or to zonation, we can try to profile in a better way also these stromal components that are differently enriched in different types of tumor samples. And we always start from the physiology. So we tried to define the transcriptional status of follicular dendritic cells, that is the core, the mesenchymal core of germinal centers, by trying to profile some follicular dendritic cell marked regions using a phenotypical marker that I showed you is NGFR, and we obtained a list of genes that are characteristics of these cells, and then applied this list to single-cell RNA sequencing datasets that are already available in the literature. But you can note that the number of follicular dendritic cells profiled by single-cell suspension are very low. So we have very few cells to use for comparison. So probing these cell types in situ is particularly valuable, because these cells that have a very branched morphology, a very stellate interweaving morphology, may be particularly difficult to isolate and to profile through conventional suspension sequencing. So based on these spatial transcriptomics approaches, we could identify some potential new markers even for follicular dendritic cells that may be used for the characterization of the associated stroma that we observe in specific tumors. One very interesting example is AITL, angioimmunoblastic T-cell lymphoma, which is a lymphoma of the germinal center in which you have a hyperproliferation of this kind of stroma, follicular dendritic cells. And indeed, the new markers that we propose in coming from the profiling highlight very well the proliferation of AITL-associated follicular dendritic cells, and may also be useful to diagnose a very rare mesenchymal tumor that is follicular dendritic cell sarcoma, for which we still need some additional diagnostic markers. So I told you that the type of stromal reaction is heterogeneous in terms of quality and also density, amount, and there are some particularly heterogeneous lymphomas. One notable example is diffuse large B-cell lymphoma, which is the most prevalent lymphoma in the Western adult, in the Western countries, in adult individuals. DLBCL might have a very diverse configuration in terms of genetics, phenotype, and also in terms of associated stromal reactions. So applying the spatial follicular dendritic cell signature to gene expression profile codes of diffuse large B-cell lymphoma, we can identify those cases that express more the FDC signature as having a predicted better prognosis as compared with those cases that do not express the same levels of the follicular dendritic cell signature. And these cases tend to have a higher enumeration of infiltrating T cells as predicted by gene expression profiling. And this is something noteworthy, since not only the classifications of lymphomas has progressively changed from morphological based ones to phenotypical ones to genetics, but now we also have some integrations to the classifications that rely on the type of stromal reactions, of the stromal architecture of the malignant proliferation. And this is particularly true for DLBCL, diffuse large B-cell lymphoma. For example, Leandro Cerchietti's group some time ago showed that we can actually define different archetypes of DLBCL according to the microenvironment composition. We have those with good prognosis or bad prognosis, and these cases with good or bad prognosis are also characterized by a germinal center-like environment in case of good prognosis, or by a mesenchymal-like state among those with still good prognosis but less immune dense infiltrate, and others with a bad prognosis are basically the inflamed microenvironment ones and depleted ones. So he defined different types of different subset of diffuse large B-cell lymphomas according to the players of the microenvironment. And some of the key players are for sure follicular dendritic cells or other fibroblastic mesenchymal cells, but also macrophages. And macrophages are another component of the stroma that is particularly interesting because we often classify them as lymphoma-associated or tumor-associated macrophages in a generic way, but probably macrophages have high diversity also in terms of their spatial localization and functional biology. Indeed, we tried to dissect the biology of macrophages residing within the germinal center and within different regions of the germinal center, like the dark zone, the light zone, and in extrafollicular regions, and try to see whether these macrophages that also show a different morphology. As you can see, the macrophages in the dark zone seem to be more epithelioid. Those outside the GC tend to be more spindle or stellate. And whether localization and transcriptional identity and maybe function could be linked together. And we performed another experiment of spatial profiling. This time we tried to define the transcriptome of macrophages residing within the germinal center, in the dark zone, light zone, or outside the germinal center, and also those that associated with an overtly developed lymphoma. So in order to see whether there were specific transcriptional characteristics of macrophages that were associated with their localization and with the factor of being already entrapped within a lymphomatous proliferation. And in order to understand a little bit more about their biology, we compared the transcriptomes that we obtained from the spatial topographical profiling of these regions with a single-cell RNA sequencing atlas of monocytes and macrophages that have been developed by a colleague, Florent Guinou, and then try to identify which specific subset of macrophage we could identify in different portions of the tissue. So to make a long story short, we could actually define germinal center residing macrophages as macrophages with a TREM2-like identity, those residing in the dark zone as those macrophages that show expression of S1, and the macrophages already associated with an overt diffuse large B-cell lymphoma more skewed towards an IL-4-induced one state. So all these types of macrophages are macrophages that have been already defined according to their immunoregulatory function, like, for example, TREM2. TREM2 expressing macrophages tend to suppress CD8 cytotoxic T-cell responses in tumors, but also in normal immunoregulatory conditions, and S1 expressing macrophages that are those characterizing the macrophages residing in the dark zone tend to be macrophages associated with the repair, with tissue repair in conditions of normal response to wound or stress, like wound healing, or in the regeneration of tissues, and still have a regulatory function, while the IL-4 induced one-like state is a recently described state of macrophages associated with an immune suppressor function, and most of these macrophages are the most abundant population observed in many tumor histotypes. So also the macrophages are a very important part of these archetypes, of these prototypes of tumor microenvironment composition, and along with stromal cells, they contribute to the diversity of these tumors and to the prognosis. Indeed, by using the characteristic signatures of the macrophages that I just showed you, so those of macrophages of the germinal center or dark zone macrophages, or those residing in DLBCL, in DLBCL tissues, we could predict the prognosis of different diffuse large B-cell lymphoma reference series. And so basically the macrophages are a relevant component of the microenvironment, or and that actually need some functional investigation. Just some few slides about another stromal context and environment. So modifications in the stroma and tumor-associated stromal changes may be either induced by the malignant clones towards the engendering of a tumor-supportive environment in the local development site of the tumor or in colonization sites, but they can also be detrimental for the hematopoietic function of the bone marrow. Here you can see the example of a myeloproliferative malignancy, primary myelofibrosis, that produced a complete derangement on the right of the hematopoietic niche toward the accumulation of extracellular matrix, overt fibrosis, and the type of degeneration that we call osteosclerosis, that is the terminal stage of the progression of this disease. But these changes are very dramatic, and some changes that are more subtle may occur and perturb the function of the primary hematopoietic function of the bone marrow in tumors, and not only in primary malignancies of the hematopoietic systems, but also in peripheral tumors. Indeed, in primary defects of the hematopoietic system, we can easily figure out how the defective cells may induce a defective hematopoietic niche that may induce ineffective erythropoiesis, altered myelopoiesis, and eventually other kinds of alterations that may configure extramedullary hematopoiesis. I'm going to define these in a few seconds. But tumor-bearing hosts means every host that has a overt developed malignant tumors in the periphery. Let's say, for example, a colon cancer or a breast cancer. Although the bone marrow is not infiltrated, the presence of a malignancy can still drive a dysfunction of the hematopoietic system and can still drive the normal differentiation process of hematopoietic cells toward derangement and alterations in myeloid cell differentiation, in erythroid cell production, and even a remodeling of the stromal architecture of the bone marrow. And at the end, we can also observe extramedullary hematopoiesis in these settings that are not associated with a primary tumor, a primary malignancy of the hematopoietic system, but rather as a secondary event that is in some way perturbing the bone marrow homeostasis. And when these conditions occur, the other niche that I'm going to describe that is involved is the vascular niche of an organ, actually more than one. The spleen is one and the other is the liver, but let's focus on the spleen, of an organ that has already harbored hematopoiesis during development. So during embryogenesis, we have a phase in which the hematopoiesis is confined to the splenic sinusoidal elements, the yellow lines that you see in this picture. And the vascular compartment of the spleen may be pushed towards hosting again this hematopoiesis that in this case is pathological. And this is a very peculiar vascular niche because it expresses a marker that is not usually associated with endothelial cells, which is CD8 alpha. Probably most of you know CD8 as a T cell associated marker, and indeed it is. It's a receptor MHC class 1, co-receptor MHC class 1. Usually it is expressed in T cells as a heterodimer CD8 alpha beta, while in the splenic endothelium it's expressed in the homodimer alpha alpha version. And on the immunofluorescence on the right, you see the colocalization of CD8 alpha with another endothelial sinusoidal marker, PLVAP. And on the left, you see the colocalization with a nuclear marker that is very specific of endothelial cells, that is ERG. And the expression of CD8 is not associated with the T cell receptor signaling machinery. So the CD8 is not playing a role, the same role probably that it plays on T cells or other immune cells. In these conditions, when we have a perturbation of the bone marrow hematopoiesis, intravascular splenic hematopoiesis can occur as a complication, and also in tumor-bearing individuals, not only in individuals with a primary hematopoietic defect or hematopoietic malignancy. And here you can see the sinusoids that are lined by CD8 in the lower central picture that are filled by malignant megakaryocytes and myeloid cells in a primary myelofibrosis case. On the right, you see the endothelium filled with malignant hematopoietic cells highlighted by CD8 and ERG. And again, now we have the tools to try to characterize in detail the modifications of this very peculiar, first of all, to understand better the biology of this very peculiar endothelial vascular niche. And secondly, to identify the modifications that this niche undergoes when a myeloproliferative disease or extramedullary hematopoiesis related with a hematopoietic neoplasm is in place. So we performed a digital spatial profiling experiment of the CD8 positive endothelial cells. We obtained a signature of these cells and validated it on single cell datasets, single cell suspension RNA-seq datasets, and these single cell RNA-seq datasets confirm that the spatial splenic signature that we obtained from the experiment was actually highlighting the endothelial cells specifically in the spleen and not other endothelial subsets. So this is normal splenic endothelial signature that is very specific of this endothelial subset and not shared by other sinusoidal endothelial or other endothelia in other cells. And through this approach, we could identify a very strong core differential signature. Some of these can be validated also by conventional immunohistochemistry, but these are completely useless because, you know, so far nobody needs a new marker for splenic endothelial cells. We have selected a lot of them, but there are some markers that are more important than others. NR5A1 is a nuclear receptor, is a transcriptional controller, and the interesting fact about NR5A1 is that it actually regulates steroid biosynthetic process there, and in the transcriptome of the endothelial cells, there is a well-defined module that is dependent of NR5A1, and it has been reported a long time ago that mice lacking NR5A1 have a defect in the splenic vasculature branching. So there might be some link between this marker, this nuclear receptor, and endothelial function. And there are patients that have mutations in NR5A1 that have asplenia as a clinical phenotype. So we decided to try to induce the expression, to force the expression of NR5A1 in an endothelial cell line that shares some feature with the splenic endothelial cells in terms of the expression of some lymphatic endothelial markers. And we could actually obtain the modulation of several genes that belong to our splenic endothelial signature, but not to recapitulate the splenic endothelial phenotype. So there's still something that is missing. Probably NR5A1 alone is not able to reprogram endothelial cells towards the fate that I showed you. Just the very couple of last slides to tell you that under the pressure of extramedullary hematopoiesis, the endothelial cells of this particular vascular niche change. And they change their transcriptome and they're reprogrammed. And among the genes that are downregulated, there is the entire endothelial, splenic endothelial signature. And most of the hallmarks of endothelial identity are downregulated, while other markers that are associated with either inflammation or with the enhanced trafficking and permeation of the endothelial cells may be induced in cases in which you have the pathological intravascular hematopoiesis associated with myelofibrosis. And we now started validating some of these markers that can be of some interest because they could be markers of an endothelium that is sensing the presence of intravascular hematopoiesis, and they might be used also to study a little bit more on the function of the endothelial cells that are deranged by the presence of malignant hematopoiesis. So I tried to drive you through different types of stromal niches associated with malignancies of the hematopoietic system, and to show you that stromal cells that are usually not actually the main actors, main players in this drama are important part of the heterogeneity and probably much more than bystanders in this setting. Thank you so much for your attention. I hope it was not. Thank you.
Claudio, thank you so much for this enlightening lecture. So I don't know if there are questions from the audience or curiosities. Yes?
Thank you, Professor, for this lecture, let's say. So, metabolically, do you find some specific pattern activating the stromal components of some specific disease instead of another one? This was my first question. And the second one, if you were able to associate a stromal component based on or germline mutation or sporadic mutation. I mean, based on one case or in another case, you can have a different landscape, let's say.
Very good points, very good questions. So first one, yeah, there clearly are different states in terms of metabolic programming of stromal cells in stromal reactions associated with the different lymphomas or also myeloid malignancies. But they are actually dependent also on the setting of the probing. You know that some tissues are more hypoxic, or some niches are particularly hypoxic as compared with others. And in these cases, the conditions in which you do this kind of measurements may impact a lot on the output of metabolic interpretation of metabolic features. But yeah, we already know that some tumors that have a higher proliferation rate or different types of OX-phos or glycolytic metabolism may have different types of influences on stromal cells. But there is no current metabolic-based classification of stromal types, of stroma reaction types in lymphomas, not yet. You know, the dominant programs are mostly related with cell adhesion or types of cytokines or chemokines that are expressed. But this is for sure very...
Yeah, since that metabolites act as a cofactor of epigenetics, so can explain why you find a transcription program instead of another one.
Yeah, for sure. Yes. But also the epigenetic part is not really well studied in this setting. And as far as the second question is concerned, you know, in some tumors in which genetics are very well defined and genetic subsets have been well defined, like in the LBCL, there are clear association between genetic subtypes and types of stromal microenvironment composition. But I'm not aware of anything regarding the presence of germline variants as compared with somatic ones in these settings, even if you know that there are some germline configurations that may impact already the basic composition of hematopoietic system. But it's very hard to uncouple the stromal defects from the associated hematopoietic one.
Thank you.
Any further curiosity?
Thank you very much. I was very impressed by your adoption of AI, but I think you showed a good combination of AI and CI, artificial intelligence, and Claudio intelligence.
Thank you.
And I have a question, a comment in the question. The comment is that when I was young, there was a kind of dichotomy between the morphological approach and the biochemical approach. And I've been, through my training, been involved with that. I think it is very gratifying that this can be integrated so effectively as you have done. And the question is, two questions. One is the macrophages. Macrophage experts tell me that there is an important distinction between the blood bone marrow derived macrophages and the so-called tissue resident macrophages. And I wonder whether you agree with that and which one you think are more relevant to your work. And the second question is the relationship between the tumor and the so-called stroma. I agree with you, it is very important and more subtle than often is realized. However, when there is a very, what we call a very powerful set of mutations, I get the impression that this sometimes is kind of overtaken.
Yeah.
Do you agree with that?
Completely agreeable. Yeah. So first question, very relevant. The tissue-resident macrophage is probably more a state than a cell type. And because under certain conditions, also tissue-resident macrophages will be renewed by blood-borne, blood-derived macrophages. And the local proliferation and local expansion dynamics, it has been also showed for Langerhans cells, for example.
So you think they're not all that different?
No, they are actually, but I mean, in the conditions in which we observe a local proliferation of a malignant clone and the initial phases of a rewiring of the local microenvironment, the resident macrophages play a key role, and most of the phenotypes that we find associated with the worst prognosis correspond to tissue-resident macrophages. And S1 positive macrophages, for example, those that contribute to tissue repair are by definition tissue-resident macrophages. What I'm saying is that I'm not sure that on the long run, all these myeloid components that are not, tumoral and may be heavily contaminated also by the influx of monocytes deriving from the periphery. So this is something that is probably more of a dynamic process, and it may be different also if probed at different stages of tumor evolution or at different tumor sites. So I cannot say whether they are more important or the blood-derived ones are, the monocyte-derived ones are more important. It might depend on the also tumor site and stage. As far as the second point is concerned, you're completely right. I mean, so here the point is the genetics are usually matching the environment composition. So whenever you have specific type of genetic lesions, you observe a certain type of tumor microenvironment composition that may be associated either with the immunogenic profile, specific immunogenic profile of a tumor. But as you said, when you have specific strong drivers, let's say, for example, double-hit lymphomas, or those lymphomas that have a MYC translocation, even alone, like in the Burkitt lymphoma, the microenvironment tend to be a depleted microenvironment, and the role of the stroma is much less relevant. So I'm not sure that this applies to all the strong genetic drivers, but for sure in some conditions in which the tumor is reprogrammed to express low levels of MHC and to downregulate most of the molecules that favor the crosstalk with the microenvironment, the role of the stroma itself and the microenvironment overall is not so important. And this is something that we observe in many types of tumors, not just in hematological malignancies. But again, also here, it's also a matter of staging and timing. There can be windows of time in which the interplay between the tumor and the microenvironment is very relevant for selection and immunoediting, and then probably there are some other stages at which microenvironment can be seen.
Good. One more.
Thank you very much, Claudio, for this very, very beautiful lecture. Just a curiosity, do you have observations on stroma reconstitution after transplantation. I mean, do you see the same niche in successful transplant after transplantation?
You mean same
Hematopoietic niche in the bone marrow.
Or normalization? Normalization of the niche or?
Normalization of the niche.
Okay. Yes, you can. This is part of a good response to transplantation. The stromal niche can be reverted by the presence of a normal hematopoiesis that takes place upon successful transplantation. But it's also true that the primary defect in the stroma can affect the outcome of the transplantation. For example, in an experimental setting like in beta thalassemia mice, in which you have the presence of a coexistence of beta thal hematopoiesis and a deranged stroma with iron overload and stromal alterations, the bone marrow transplantation with wild-type hematopoietic cells tend to fail because of the presence of a stroma that is already primed by the beta thal cells that result more fit in that environment as compared with the wild type. But if the wild type makes it, and if the reconstitution is achieved, then the stroma will normalize as well. So the stromal niche can be actually normalized by successful transplantation.
Excuse me, you mean that in leukemia, even if you make a retrospective study, in leukemia relapsing after transplantation,
relapsing, okay.
Immediately after the transplant, the reconstitution is normal in terms of niche?
No, okay. So if you mean relapse immediately after transplantation, you might still have some defects.
Even relapsing after 1 year. I mean,
it's
if you have any observation on this follow-up.
It's very hard to say because besides fibrosis, you don't have much to measure. So you might have some alterations in the phenotype of mesenchymal cells, but not much more. For sure, in those with fibrosis, the chance of relapse is higher, actually. Yeah.
So maybe I can close with one more question. That's actually two. So you pointed out at the beginning when you do these experiments where you transplant a lymphoma and you look at different organs, and you see basically that apparently there is also clonal selection of the clone in the different in the different elements. You didn't point out the effect of spark knockouts in this context, but basically I understand that the message is that the stroma has an impact on that clonal selection.
Yeah.
So how do you actually interpret this result? So how does the stroma influence the clonal selection? And is this then a genetic selection? So namely the clones in the different tissues have, variants due to the heterogeneity of the lymphoma, or is it an epigenetic adaptation?
Okay, so we actually know just part of the story. We know that, for example, in the presence of a defective extracellular matrix, like in spark defective hosts, you have a better tendency to T-cell activation, CD8 T-cell activation, and so you exert a higher immune pressure over the engrafting clones. So this is the reason why you have different compositions between the spark knockout and the wild-type mice. As far as the differences among different sites are concerned, we actually don't know. We know that different mutations are also selected in terms of fusion transcripts. And since this is RNA sequencing, we could not just infer mutations from RNA-seq, or we just looked at the fusion transcripts over there. And there is a different selection. But if the selection is actually something active and driven by the environment, or just by chance, you can't even say. What we know is that the initial composition of the tissue is determining the type of the stromal reaction that you will see in the tumor. Yeah.
And then the other question relates to clonal hematopoiesis.
Yes.
So have you ever looked at the impact or the correlation between the extent of clonal hematopoiesis and the restructuring of the stromal compartment in the bone marrow?
No, we didn't. So it's we still lack a good tool to localize in situ clonal hematopoietic events, besides some that are actually, JAK2 mutation. You can look at JAK2 mutant or P53 mutant cells and then try to infer something about the surrounding stroma. But we still don't have this fine. In the case cohort that we studied with Cornell based on MDS and AML cases, there were also some CHIP cases. And in those cases, we still miss the stromal architecture analysis. So we have to integrate also data about the stromal composition. I would not expect much in terms of because they are pretty scattered and they are part of the ALIP clusters as much as other non-chip bearing mutations. But it would be very interesting. Yeah.
Okay, so if we are all happy with the questions, thank you so much again, Claudio. Great contribution. Okay, so I guess we have to move on with our short but very precious talks. I think there is a list here with a priority that goes following the list. So I would ask Stefano to join us and update us on his projects. So the title of his short talk will be Chromatin Networks Unveils ATF3 Role in Sustaining Bortezomib-resistance in Multiple Myeloma. Thank you, Stefano. So let's try to be as sharp as possible with time.
I'll try.
Yeah. Hang on, we just have to set. So just while we're waiting, just to also, for sake of transparency, so we asked every member of our community, of our NAC community, to provide a title and an abstract. And due to shortage of time, we had to select few, but everyone will be represented in the next years. We look forward hearing to all the different contributions. So I apologize for those of you who will not speak today, but you will have a chance next year.
Okay. Okay. So thank you so much for the opportunity to let me talk about my PhD work that I'm carrying at the National Cancer Institute Regina in Rome and the University Sapienza. And I'll talk you about the chromatin networks and how we applied them to analyze the Bortezomib-resistance in multiple myeloma. So first I want to talk briefly about cancer evolution and epigenetic cancer evolution, and we can imagine and picture the cell development as a surface with some hills, and cells can take some path, the simplest path through development. However, this surface can be shaped thanks to some epigenetic alteration, so chromatin alteration. And what happens is that these hills are shrinked, and a cell can move back or forward and move along different developmental path. And this is very important in cancer because cancer is not a unique group of cells, but it's composed by different subpopulation of cells, and some of them can bear some epigenetic alteration that give plasticity to them, and in this way they are able to shape the transcriptional landscape to adapt to external stimuli such as the therapy, and some of them are able to resist, so then can re-expand and give relapses. And epigenetically talking, some of these regions are named the cis-regulatory elements. So all the region, the non-coding region that are able to define a transcription landscape through space and time are regions such as enhancers, that are this one, the orange one, or promoters, or some other type of non-coding regions. And what we do in our lab is to apply these concepts to multiple myeloma. So multiple myeloma is a hematological disease occurring in the bone marrow, in which plasma cells undergo a clonal expansion and produce a very high amount of immunoglobulins. And this is a very heterogeneous disease, both genetically and epigenetically. However, some works identified specific epigenetical modulator, let's say some transcription factor or transcriptional regulator that are shared among patients. And so our question is, are we able to define some epigenetic determinant, some cis-regulatory elements that are involved in multiple myeloma proteasome resistance? And proteasome is a very largely used treatment for multiple myeloma, that is a proteasome that target the proteasome, and that are multiple myeloma cells really need the proteasome due to the high production of proteins. So to do that, we analyzed a large cohort of multiple myeloma patients, a longitudinal cohort of 44 multiple myeloma patients from 4 different stages. The MGAS, that are the pre-lesion samples, 7 MGAS, 13 pre and 13 post-treatment patient matched pre and post-treatment patients, and then 11 relapses. We applied a method developed during my PhD, that his name is Ticronet, that create a co-accessibility network, a chromatin network, in which nodes are regions, detected regions, and edges represent the correlation in the accessibility signal along all our cohort. And we applied some analysis on this network to stratify our accessibility regions and define a group of 2,200, more than 2,200 regions. And what we obtain is showing the signal of these regions. In our cohort, we were able to divide our cohort into 2 clades. The first one that is mainly composed by disease-like patients, and the second one that is mainly composed by healthy-like patients. But the most important thing is that we are able to split our post-treatment, that are the white one, into 2 groups. One group that has this region completely closed, and another one that still bears some accessibility signal on these regions. Despite for clinicians, they are completely disease-free. So we analyzed, we focus on these regions, and also matching the same patients before and after the treatment, we saw that we have the same trend. So the signal is very high in all the pre-treatment and decrease in some post-treatment, but not in a small group of them. So we decided to investigate the regulator of this region. So if we have an accessibility region, we also have some transcription factor that can bind them. So we applied a footprinting analysis of all the known transcription factors, and we focused on this group of transcription factors that has footprinting mainly reached on these regions and are reached both in disease-like and also in some post-treatment samples. To validate this transcription factor, we applied a gene regulatory network analysis on primary multiple myeloma cells from our lab, divided into 3 groups: the T0, so the sensible one, and then RS1, RS2, that are the same cells that are resistant to multiple myeloma to different concentrations. So applying gene regulatory network analysis, we obtained that ATF3 is the only transcription factor that we found both in the footprinting analysis and also increased its activity, since this is the upper right quarter, increases activity both in RS1 and RS2, the resistant cells, compared to the sensible one. And also we defined the target gene through a ChIP-seq experiment of the ITF3, and we saw in the same single cell experiment that the sensible cells are have a lower expression of these target genes compared to RS1 and RS2. So this means that ATF3 could be linked, according to our analysis, to resistance path. So we did some experiment in lab, and we saw that actually there is an higher activity of ATF3 in the resistance cell, and there is also a higher expression of ATF3 compared to the sensible one, and also the protein is higher in the resistant compared to the untreated, so the sensible. And also, I'm sorry about for the pictures, but we also saw that ATF3 is increased. Here we analyzed some patient samples before and after treating the patients with the samples with 5 nanomolar of bortezomib. We saw an increase in the expression of ATF3. According to our analysis, we detected ATF3. Here is the promoter, the box here, but also in the 2,200 regions, we also detected 2 regions here, this one and this one, that are putative enhancers of ATF3, and here represented from a CHIA-PET experiment, the link between these regions and the promoter. And interestingly, these regions, same here in the same patients, treating the samples with bortezomib, the expression, so the RNA production of these regions increases also for the both two enhancers. And again, we tried to interfere ATF3. So what happened if we interfere ATF3 and we treat the cells with bortezomib? We saw that there is a decrease in the IC50 of sensible cell if we interfere ATF3 compared to the control. Here we did the experiment in KMS 18 that are sensible cell, but we replicated also in resistant cells, and we saw the same decrease. The violet line is the interfered and the blue one is the control. So we see a decrease here in the IC50, also in the resistant cell. So we know that we detected ITF3 is a putative regulator in some ways of the resistant in multiple myeloma, but since transcription factors are very hard to be targeted, we decided to identify some target gene that we can target and use for like a therapeutical target. So we analyzed a single cell experiment, a public single cell experiment of 18 patients divided from into optimal responders and suboptimal responders to bortezomib. These are single cell RNA experiment. We took our genes before the target of ATF3, and we applied a machine learning method to select the features. And what we obtained is a group of 86 genes that increase their expression in suboptimal responders compared to optimal responders. But the more important things that we found is that if we divide the patients according their clinical annotation into very good partial responders, stable disease, and progressive disease, we see that the expression of these genes follow a trend in which their expression is higher in patients that respond worse to the bortezomib. So actually we think that ATF3 and the gene regulated by ATF3 can be in some ways involved in Bortezomib resistance. So we took these 86 genes, we applied a protein-protein interaction network analysis, and we saw that these genes are mainly involved in some known pathway of Bortezomib resistance, so the proteasome blockade resistant processes such as unfolded protein response and cellular response to stress. So actually, we are pretty sure that ATF3 is involved. The most important thing is there are very few papers on ATF3 and Bortezomib, and also let's say there are few papers on ATF3 and it's involved in Bortezomib. So just to be very brief, this is still an ongoing project. We are working on it. We are trying to detect the target gene and validate them. But however, we were able to detect a subset of regions, of epigenetic regions that are modulated in our patients according to the disease-like or LT-like definition. And then we defined ATF3 as a putative regulator of Bortezomib-resistance and the cancer evolution in multiple myeloma. And also, we detected that interfering ATF3 can decrease the IC50 of our cell, showing that ATF3 is strongly related in some way to the answer of multiple myeloma cells to Bortezomib. So that's it. I thank you for your attention, the opportunity, and also all the people that worked on this project in Rome and in Vienna. So if you have any question, I'm very happy to answer now or later when you want. Thank you so much.
We have time maybe for one question.
If the resistance is due to an epigenetic event altering the expression of ATF3, have you tried to add a small amount of azacitidine?
Scusate, se non ha il microfono il professore non sentiamo, quindi dategli un microfono. Chiunque fa domande. Grazie.
You heard me, but I will repeat for you. So if the change in ATF3 is due to an epigenetic phenomenon, then azacitidine, known to interfere with epigenetic events in small dose, could cooperate with bortezomib.
Okay. I think that the expression of ATF3 and also the enhancer that we found are not directly related to epigenetic alterations such as acetylation, histone acetylation, because we saw that before and after the treatment, we have the same signal of acetylation, for example, in enhancer and in promoter. So what I think is that bortezomib in some ways activate some other transcription factors, such as one of the known is NFKB, that we saw that there is some pathway, the non-canonical pathway that can regulate ATF3. So we think that ATF3 in some ways activate genes involved in the resistance, but is not epigenetically, at least in this case, not epigenetically regulated.
Okay, so I guess we have to move on. Thank you very much again, and
Thank you.
congratulations.
So, hello everyone. Today I'm going to show you some new data of my PhD project. The title of presentation is Exploring the Impact of Pathways. So basically we are going to talk about T-cell dysfunction in the context the T-cell dysfunction in solid tumor is induced by the cancer itself, but also by the tumor microenvironment, who suppress the T-cell function in different ways, so such as immunosuppressive cells, or the release of immunosuppressive cytokines, the persistent inflammation. And another key point is the repeated antigen exposure who allow the establishment of this type of dysfunctional T-cell state known as T-cell exhaustion. So the T-cell exhaustion basically is the loss of the whole functionality of the T-cells, such as the decrease in cytotoxicity, in cytokine production, proliferation, and also in the amount of receptor at the cell surface. But this point we will understand better later. So one of the hallmarks of T-cell exhaustion is the expression on the plasma membrane of the T-cells of this receptor known as checkpoint blockade, such as PD-1, CTLA-4, who allow the deactivation of the T-cell and the loss of T-cell functionality by the binding with immunosuppressive ligand, PD-L1, PD-L2, and the other. So the traditional cancer immunotherapy has been focused for many years to the targeting and blocking of these checkpoint inhibitors. But the last year, a new target has been developed, and in particular, the intracellular signaling protein of T cell, and this is the acylglycerol kinase family protein, which are lipid kinase who convert the acylglycerol into phosphatidic acid and allow the shutdown of the signal in the T cells. So basically, there are the different type of isoform of this acylglycerol kinase, but the two mainly expressed in T cell are the alpha and the zeta isoform, but the alpha one is the one in which I'm involved in this project because convert a small pool of the acylglycerol at the immune synapse. Okay, so basically, it was discovered as a negative regulator of the T cell signaling from genome-wide CRISPR screening in primary T cells. A point that I want to stress is the differential expression of this protein compared to the other checkpoint inhibitors that we were talking before. And in particular, when T cell is functional, there is a complete T cell receptor activation that inhibits the expression of the protein. While when we are in a dysfunctional state, there is a partial TCR activation, and this leads to of the protein. So basically, the differential profiling compared to the other checkpoint is because it is upregulated in a naive T cell, then undergo downregulation upon activation of TCR, and then the protein is highly expressed in the dysfunctional state, while the other, upon activation, increase the level and then goes up. So another key point is that DG-kinase alpha has discovered from our lab to be a protein that controls the T cell receptor dynamics. More specifically, physiological, the T cell receptor is internalized and then either recycled or degraded. In the experimental condition in which we were working on, without subprotein, which is an immunodeficiency with a loss of function of subprotein, there is a deregulation in the TCR trafficking with an increase in lysosomal degradation. But interestingly, in this project, we observed that by targeting both SAP and DG-kinase protein, we reestablished the amount of T cell receptor at the cell surface. So we observed an increase in the recycling rate. For this reason, and based on this data and the other that I was talking to you before, the biological question of my PhD is there evidence that the DG-kinase alpha mediates mechanism driving T cell exhaustion? And the answer is that several studies has shown that there is an increase in mRNA protein level in different type of cancer and with different type of T cells. So tumor-infiltrated lymphocyte, TCR edited T cells, and also with CAR T cells. Here I'm showing you only one that correlates the protein expression, the amount of protein expression in tumor-infiltrated lymphocyte with the tumor stage. So basically in this work they show that by targeting both PD-1 axis and DG-kinase alpha, there is an increase in percentage survival of the mice and a decrease in the tumor volume. So in order to understand which are the molecular mechanism behind this synergistic effect, I developed an in vitro system to mimic the T cell exhaustion phenotype, and we use primary T cells that is stimulated by cancer cell with transduct with OKT3 single chain variable fragment, which is an agonist of T cell receptor and allow the stimulation and activation in MHC independent manner. The choice of different cancer cell line was based on screening of the expression of immunosuppressive ligand PD-L1, 2, and AIM-CH class 2 receptor. And we choose pancreatic cell line, breast cancer cell line, and melanoma cell line in order to stratify the expression of this immunosuppressive ligand. We, of course, developed a protocol to editing primary T cell from healthy donor, and all the result that I'm showing you is with the different healthy donor. So basically, I was able to set up an in vitro assay for chronic antigen rechallenge, in which we stimulate in chronic the same T cell, of course, wild type and digikinase alpha knockout. And then we evaluate the exhaustion marker at different time point and stimulation. So what it was clear is that there is an increase in both the condition during time and stimulation of the PD-1 positive cell, CTL4 and LAG3. But interestingly, without digikinase, we see there is an increase in percentage positive cell of PD-1, CTL4 and not LAG3. So then this experiment was made in effector ratio 1:1 with breast cancer cell line as target. So then we moved to melanoma cell line in 1:10 effector target in order to stress a lot this primary T cell and repeat the experiment. In this case, we measure also the killing activity, and we can appreciate again in the killing activity, but not any difference in the expression of exhaustion marker at the plasma membrane. So again, we repeat the experiment with 1:20 effector target. There is always a gain in killing activity and always no differences in the expression of exhaustion marker. Then we try with another donor the same experiment with all the different cell line, and we can appreciate that the gain of killing function was measured upon chronic stimulation. So the targeting of digikinase is give a gain of functionality to the T cells only after the third stimulation. At least with pancreatic cell line and melanoma cell line, but not with the breast cancer cell line. And this may be, but we have to do a lot of much investigation, is due to the expression, the high expression level of PD-L1 or PD-L2, or maybe the cell itself are a little resistant to apoptosis. So also in this case, we evaluate the exhaustion marker, and there were no differences, meaning that we are uncoupled the killing functionality of the T cells and the fitness of the cells from the expression marker at the cell surface. So in conclusion, now we want to go in the deep of molecular mechanism behind this synergistic effect between the PD-1 axis and the alpha knockout or overexpression of the protein. So in collaboration with Prof. Tom in Lausanne, they are sending us their construct. These are Jurkat cell line carrying the PD-1 construct wild type, of course stimulated and activated by Raji cell with PD-L1 and PD-L2. Then another Jurkat cell line carrying PD-1 double mutant in the tyrosine that are required for the recruitment of SHIP1 and 2 that are phosphatase who inhibit the T cell signaling, and then the last, the Jurkat cell line knockout for SHIP1, 2, or both. So the conclusion is that we want to understand whether there is an enhance in T cell signaling following PD-1 activation in digikinase alpha knockout, or a decrease in digikinase alpha overexpressing cells, and then move to understand also the TCR dynamic upon and following the PD-1 axis activation. So thank you for your attention and for the opportunity.
Time for one question. So maybe I can ask you, I think the obvious one, which is, have you checked the TCR levels in this killing, in this different context? Are they gaining the surface level of TCR?
No. At the last day of stimulation, there is a little decrease of the amount of TCR in an increase in knockout cell line, knockout for digicanes compared to the wild type, but it is very variable because the experiment it's long and the experimental setting is not easy to measure also that point. So for answer and show the data, I have to repeat.
Okay.
So as preliminary, I can say yes, but.
But is the increase in TCR expression something that happens right away in the knockout from the very beginning, or only upon stimulation you keep longer or higher level of TCR?
No, also basal.
Also basal,
yeah.
okay. Okay, thank you very much, Alessia. And we go now to the next, Giorgia Silvestrini. Thank you very much, Giorgia and Alessia. So Giorgia I heard that you were not feeling so well. I hope you're doing better, or? No, pensavo che stessi a casa oggi, non potessi. Okay, so her talk will be deciphering the stroma-aml cell crosstalk in an in vitro study model to unveil the background mechanism of extramedullary progression in acute myeloid leukemia. Thank you.
Hi everyone. I am postdoctoral fellow at the Oncohematology Laboratory, headed by Professor Boso at Vergata University, and my research project is focused on the characterization of extramedullary acute myeloid leukemia. No? It doesn't work. This one? Okay. Okay, first of all, I will introduce to acute myeloid leukemia. Acute myeloid leukemia is an heterogeneous hematological malignancy characterized by the clonal expansion of undifferentiated myeloid precursor cells, then blasts, leading to an impairment of hematopoiesis and bone marrow failure. This occurs because neoplastic transformation alters the mechanisms that regulate the proliferation and differentiation of the hematopoietic stem cell, preventing its maturations. And this leads to the accumulation of leukemic blasts first in the bone marrow and later in the peripheral blood and other organs and tissues. When leukemic blasts migrate outside the bone marrow niche, patients experience a more uncommon manifestation of acute myeloid leukemia, referred to as extramedullary acute myeloid leukemia or myeloid sarcoma. Now we are focusing our attention on the role played by bone marrow microenvironment, particularly mesenchymal stromal cells, as it is reported that the level of bone marrow niche pro-inflammatory microenvironment is established where leukemic cells sense and experience a variety of signals from tumor stroma, and as a consequence they acquire a more aggressive phenotype, increase cell proliferation, and become resistant to apoptosis and acquire drug resistance. In particular, our experimental plan involves direct contact co-cultures between our in vitro model, that is OCML3 leukemic cells, and mesenchymal stromal cells freshly isolated from acute myeloid leukemia patients and healthy donors. We proved that the presence of EML-derived mesenchymal stromal cells, as you can see in the upper left figure of this slide, increase the capability of migration of leukemic cells as compared to cells in monoculture. And this confirms the capacity of mesenchymal cells to support the EML progression. Moreover, treatment with metformin, that is a drug capable of inhibit tumor progression in several cancer models, decrease the number of leukemic cells both in co-culture and monoculture. However, the presence of EML mesenchymal cells are able to mitigate the effect of drug treatment. The same findings also apply to the cell invasion assay. When it comes to co-cultures with healthy donor-derived mesenchymal cells, we can appreciate the fact that healthy donor mesenchymal cells significantly supported the migration of OCML3 cells, but in the case of invasion assay, co-cultural OCML3 cells are not capable of invading the HCM coating. So the left panel shows confocal microscopy images related to our co-culture system because we choose to depict the aspects related to cell-cell interaction and in particular observing co-cultures under a phase contrast microscope. We observed that a proportion of OCML3 cells remain adherent to the mesenchymal substrate. So using a dye tracing approach, we labeled mesenchymal cells with DID and OCML3 cells with dye that are lipophilic tracers that are well retained by cell membranes without affecting cell viability in any way. In particular, we observed that in the case of treatment with metformin, we noticed that there was a reduced number of leukemic cells adherent to the mesenchymal cells in both conditions, and that there was a huge number of leukemic cells adherent to the EML-derived mesenchymal cells as compared to healthy donor-derived mesenchymal cells. To confirm these findings, we performed the experiments using an 8-well chamber slide with grid, which allow us to obtain a quantitative assessment of the observed phenomenon. And these are images of the related to quadrants selected for the cell count for both healthy donor and EML mesenchymal cells. First of all, we also highlighted cell-cell interaction through a 3D reconstruction of leukemic cells interaction with mesenchymal cells. And as you can see on the right panel, OCML3 cells share dual yellow and red signal that is a proof of the capability of mesenchymal cells and leukemic cells to communicate with each other. Then we counted the OCML3 cells adherent to the surface area of mesenchymal cells, and we found that an increased number of OCML3 cells adhere more tightly to EML-derived mesenchymal stromal cells as compared to healthy donor. Last but not least, we performed a phalloidin staining in order to assess the eventual occurrence of cytoskeletal rearrangement in mesenchymal cells, and we can conclude that the presence of leukemic cells does not induce any detectable alterations in terms of fiber content in mesenchymal cells in direct contact with leukemic cells. Thank you for your attention. I would like to thank my lab and all of you.
Questions from the audience?
Thank you so much. So, have you noticed any difference in the differentiation capability of the mesenchymal cells isolated from the patients?
No, we are not studying this aspect. We are focusing on trying to find difference between the kind of interaction between OCML3 cells and the two types of mesenchymal cell layer. We are performing RNA sequencing in order to highlight different in the transcriptomic profiles.
Maybe I can ask you one more thing. So can you speculate on what could be the crosstalk? So what type of molecules could potentially favor the adhesion?
We believe that in the case of interaction of OCML3 with DML-derived mesenchymal stromal cells, we believe to find the involvement of different or a differential expression of adhesion molecules in respect to healthy donor mesenchymal stromal cells, or the expression of genes related to the epithelial mesenchymal transition or marker of tumor progression.
Okay. So if there's no further question, then thank you so much again. Adesso. Aspetta un attimo.
What about microvesicles?
It's one of our future plans to isolate extracellular vesicle from mesenchymal cells in order to study the presence of miRNAs that are up and down regulated in our cohort of mesenchymal cells from patients.
Good. Thank you very much. Then. So we should be now connected to Enrico Attardi, who is actually not with us today, but listening to us from abroad. Let me just check whether we are connected. So Enrico, we will turn on the presentation here and you will ask us to move on with the presentation. Is that fine? Posso premere io? Posso andare io avanti con il telecomandino? Ce l'avete voi. Ok. Possiamo cominciare? E non c'è la presentazione. Ok. So Enrico, can you hear us?
Yes, I can hear you, but I cannot see the presentation.
Non vede la presentazione.
Okay, now yes. Okay, I will start, okay?
Hold on, noi non vediamo la presentazione però qua. Sì, questa è. Ah, ok. Enrico, we can start.
Okay, so good afternoon everyone. I apologize if I could not attend in person. This is such a great meeting. I would like to thank Donatella and the scientific committee for giving me us the opportunity to share our data about the impact of clonal hematopoiesis across glomerular syndrome and leukemia predisposition. So I think that right now you are watching the current classification of glomerular failure and leukemia predisposition syndromes as reported in 2022 in these two important classifications. This is really essential to us because it's going to confer the importance for certain patients of harboring a germline predisposition towards myeloid neoplasms. And the fact that we don't have so many differences between bands means that we already proved how could be strong to evaluate in some such specific settings the germline predisposition. Move on. So, as you can see here, at the beginning we thought that germline predisposition can only affect pediatric patients. Right now we have the proof that actually they can affect the individuals across the lifespan, and after especially the discovery of DDX41, in which the incidence of myeloid neoplasms is around 70 years old, so as the same as the classical myelodysplastic these syndromes, we now have the real evidence that this kind of disorder can affect all ages. So even as an adult hematology, we have to be aware about the fact that there's a certain percentage of patients that they can harbor this familiar predisposition. Is ahead. Okay, so now today we are going to focus on the clonal hematopoiesis that can be present in such specific mutations. So as you can see from the red round, these are different kinds of germline predisposing syndromes. One of them is most recognized. But with the blue round, you can see that many of them can actually have different kind of cytogenetic alteration and also somatic mutation. And some of them can be more or less specific to a specific disorder, germline disorders. So in a way in which we used to call them clonal hematopoiesis in predisposed individuals, because it's the haptens of the stem cell to escape from the cell fitness constraint. And so trying to have a proper clonal cell differentiation and maturation. So today I'm going to show you how, in the clinical perspective, this clonal hematopoiesis can be really helpful for us in order to recognize properly these patients, but also to stratify the follow-up and the proper treatment strategies. So they are specifically important, as I told you, for the diagnosis. And if you go, please can move ahead, you can see that there are specific disorders like monosomy 7 that seems to be more recurrent in certain bone marrow failure syndromes. It is reported, for instance, that 50% of pediatric patients harboring a monosomy 7 can have a GATA2 deficiency or SAMD9 and SAMD9L syndromes. So it can be really important, at least as a red flag, to suspect this kind of disorder. Okay. But they can be also important for some other specific difference. But before showing these, I have used the example of SAMD9 and SAMD9L syndromes, because in this specific setting, as I said, we can have the cell fitness constraint related to the germline predisposition. In this specific context, we have a gain of function of SAMD9 or alternatively SAMD9L that impacts the cell differentiation and maturation. So what happened is that, considering that our cells, hematological cells, are really dynamic, what we can have is that the occurrence of somatic mutation across different cell lines that can counterbalance the gain of function mutation. So we can have a monosomy 7. That means that we're gonna lose the germline predisposing variants, and we cannot even see it in the bone marrow or peripheral blood. But we can also have some somatic genetic rescue that is called like a sort of natural gene therapy, and that can be a second site of somatic mutation, or also a uniparental disomy of chromosome 7q. That means that can be benefit in a way in which we're gonna progress towards maturation and the differentiation. And if you can go ahead, I'm going to show you that actually the second site mutation in SAMD9 and SAMD9L syndromes can affect 64% of these kind of patients. And considering that we are not able to look at them, this mutation across different kind of leukemia predisposition syndromes, this means that this somatic mutation has the positive predictive value of 100% of having a SAMD9 and SAMD9L syndromes. And this, in terms of clinical practice, can be really helpful, because many times we have germline variants of uncertain significance. So if you have a second bit as a somatic mutation, we can for sure predict the pathogenesis also of the germline variants. But moving forward, if you can go ahead, we are going now to show the importance of the molecular monitoring of this kind of patient using the clonal hematopoiesis. Why is it so important? Because we don't have any consensus for surveillance of patients of bone marrow failure and leukemia predisposition syndromes. And this is because we don't have any recommendation right now to how to follow properly this patient before the leukemia transformation. For sure, we know that bone marrow evaluation is really important because we can check the cellularity, the dysplasia, and the blast percentages. But also the timing in which we have to perform this bone marrow evaluation across the different type of bone marrow failure syndromes actually can differ because we have low risk leukemia predisposition, but also high risk, in which we used to do this every single year. But again, we don't have any evidence for this, and leukemia occurrence can onset also before the bone marrow evaluation. If you can go ahead, I'm gonna show you that also complete blood count can be really important. But again, the complete blood count cannot predict efficiently the leukemia transformation. Evidence in the Schwachman-Diamond syndrome and IRCCS2 showed us that TP53 mutation can be present even in conditions in which we have stable complete blood count. Please go ahead. So, for this reason, what we did is a comparative analysis of bone marrow and peripheral blood across a cohort of 108 patients of bone marrow failure syndromes and leukemia predisposition in St. Jude Children's Hospital, in order to evaluate if we are able to check the clonal hematopoiesis, the CHIPI, in peripheral blood and use them to proper diagnose and track them over time. So what we did is evaluate the NGS analysis and SNP array in both sources, bone marrow and peripheral blood. And as you can see on the right, we divided the patients as inherited bone marrow failure syndromes, the acquired aplastic anemia, but also other kind of patients that can have secondary cytopenia, excluding the most common origins of the cytopenia. So, as you can see from the right, most of them, like at least 50% of inherited bone marrow failure syndromes, harbor a clonal hematopoiesis. And going ahead, if you can go, okay, that's perfect. You can see here the difference between the sources bone marrow and peripheral blood of NGS analysis. So what we found is an agreement of 91.9% of bone marrow, paired bone marrow and peripheral blood across our cohorts. And as you can see in details from the star codes on the left, you can see that basically most of the mutation appeared in both bone marrow and peripheral blood, and just few of them were exclusively found in the bone marrow, but more importantly, it was interesting to find that some others were exclusively found in the peripheral blood. And from the right, you can see that actually we have a pretty good concordance also about the variant allele frequency across this mutation. So it means that increasing the variant allele frequency in bone marrow, we also have an increase of the same one in the peripheral blood. So it seems that NGS for the peripheral blood is a robust source for detection of clonal hematopoiesis in this setting. Going ahead. Now I'm going to show you the SNP array. As you can see here, we didn't find so many cytogenetic alterations using the SNP array. It means that there are less frequent compared to the somatic mutation. But interestingly, we found across these 19 genomic alterations, most of them were found in both bone marrow and peripheral blood, but actually 4 of them were exclusively found in the bone marrow. And considering that 2 of them were the monosomy 7 and another one is a complex karyotype, we cannot say that we can use the SNP array in peripheral blood to properly track over time this kind of cytogenetic alteration. This can be really important if we are considering that in certain circumstances monosomy 7, for instance, in some benign and some benign-like syndromes, can be transient over time. So we have to detect it before proceed, for instance, to the allogenic stem cell transplant. So we have to continue to use the bone marrow evaluation. Okay, so I'm going to show you now an example of this. As you can see here from the right, this is an example of some benign and some benign-like syndromes, specifically some benign syndrome, in which the monosomy 7 in this patient appeared and remained positive over time, but changing actually the clonal sites. And on the other side, if this is gonna happen in the bone marrow, as you can see from the red dots, in the peripheral blood, you can see that it's going to decrease over time, even if we are increasing the clone size in the bone marrow. This means that peripheral blood is not affordable for this, and means also that we used to continue to use the bone marrow as a set. Going ahead. Instead, this is another example of diamond-black anemia that actually the risk of leukemia progression is reported in literature is not so high. But what we found in this specific patient was a SIBO mutation that is a clonal hematopoiesis that can progress toward leukemia. So we decided to track this mutation in both bone marrow and peripheral blood in different time points, and as you can see from the left, it seems that it remains stable over time, but most importantly, we were able to track it using the peripheral blood. That means that maybe we can continue to use peripheral blood evaluation for this specific condition, avoiding bone marrow, especially for patient compliance, because it can be painful, especially if we are doing this for a very long time period. Please go ahead. But now, if you can click again, I can show you that there are some specific situations that are called as a maladaptive clones that can be really helpful to detect them properly on time, because we can make some clinical decision on time. So if you can go ahead, I'm going to show you these 3 different examples in which we found, for instance, here in a Schwachman-Diamond syndrome, 2 different mutations in TP53 in peripheral blood. So we decided to track them over time, and in a period less than 6 months, you can see in the last time point that a TP53 mutation doubling its variant allele frequency, also with a new occurrence in with using the SNP array of the uniparental disomy of chromosome 17p. This means that the patient was progressing to leukemia without any complete blood count abnormalities, and for this reason, considering this evidence, my colleague decided to transplant the patient on time. Going ahead, we have a second example of IRCCS2. Again, you can see that we have a TP53 mutation that we track in the by the triangles using the peripheral blood. We say, okay, this can be really high-risk mutation for leukemia transformation. So it's the time to perform again the bone marrow. We did it. We repeated after 5 months, and what happened is that we found mutation in NPM1 and GATA2. That means that the patient was progressing towards leukemia, and it was transplanted as well. And moving forward, we have the third example, that is a severe congenital neutropenia. Consider that most of them used to remain stable over time. But in this specific case, what we found was a RANX1 mutation of this specific patient using the peripheral blood. And RANX1 seems to be like a second hit for this kind of specific disorder. So we decided to perform the bone marrow, track it over time in a very short period. And what we found, again, was an increase of the RANX1 mutation along with any other kind of somatic mutation that can be interpreted as a leukemia transformation. So if we can move ahead. What I would like to show you that is that basically we can use the bone marrow and peripheral blood alternate in alternative way, and maybe if we're gonna track some high-risk clones in the peripheral blood, we can absolutely do the bone marrow instantaneously and see if the patient is progressing toward leukemia. Again, why it is so important? Because we want to avoid unnecessary transplant procedure, so too early, but we would like to transplant this kind of patient at a specific time point. No, so not too late, because if it is too late, if we are in the setting of secondary acute myeloid leukemia, our data right now are showing that we have a poor outcome. We cannot benefit in the same way as performing this kind of procedure on time. So tracking this molecular activation can be really helpful to find the proper window to transplant this kind of patients. And moving ahead, we have the conclusion. It seems that prepreg-based genomic surveillance serves as a practical addition for molecular monitoring, especially in which situation where we are not able to perform, or maybe we don't have any a good compliance for the bone marrow evaluation. And the demonstrated ability to detect and track high-risk lesion underscore the potential and proof to establish new surveillance strategy for bone marrow failure syndromes and leukemia predisposition. And moving ahead, I would like to thank all the single colleagues that takes part of this work. I would like to thank Professor Maria Teresa Boso for giving me the opportunity as a visiting fellow in St. Jude's Children's Hospital, all the colleagues that takes part of this work, Marcin Wlodarski, and of course all of you for your attention. I'm happy to take any question.
Thank you very much for this enlightening presentation. So, is there any question for Enrico? Yes.
Very good, Enrico. Thank you. A short question on SAM-D9, SAM-D9L. You showed very nicely that more than 60% of these cases when monosomy 7 appears, they show a genetic event that in some way neutralizes the monosomy 7 effect. But what happens in the remaining 40% of monosomy 7 in this syndrome? Do they inexorably go to leukemia, or do you see any event, any additional event taking back the clone?
Thanks for this question. Thanks, Professor, because this is really interesting, and we are trying right now to elucidate this. Because first of all, we believe that we are underestimating the somatic occurrence in this setting. Now we are performing a specific type of NGS analysis which is called duplex UMI, in which you can find subclones even with a lower variant allele frequency, because the idea is that you don't need to completely revert the germline mutation, but sometimes it can be necessary or sufficient, even a small clone, to establish a proper clonal hematopoiesis, and so a proper cell maturation and differentiation. So the first point is that we need to elucidate in which context these specific clones can positively affect the cell maturation and differentiation. Second point, maybe we are underestimating some back mutations that sometimes can happen, because we have some specific situation in which we don't have the germline mutation with a VAF as 50%, but we are not able to check other kind of second site mutations. And the third point is that now we are also focusing on the fact that some specific somatic mutation can be compartment specific. That means that they can affect the myeloid compartment, but sometimes also the lymphoid compartment. And this can be really helpful potentially for this kind of patients, because they also have immunodeficiency. That means that maybe with the somatic genetic rescue, we are able to reset lymphocytes function and surveillance in a way in which we can decrease the bone marrow inflammation. And I really believe that for instance, comparing SAMD9 and SAMD9-like syndromes with GATA2 deficiency, actually the risk to leukemia progression is lower compared to other kind of leukemia predisposition syndromes. So maybe the inflammation can have a major role for the bone marrow failure syndromes, and maybe, again, it's just an hypothesis that we are trying now to go further to this, but maybe the lymphocyte compartment can play an important role to decrease this kind of inflammation and so reduce the severity of the cytopenia.
Any other one? So maybe I have just one that is more a curiosity, Enrico. So I think this adaptive responses that the stem cell has, or the progenitors have to the original mutation, as you pointed out now, have also an impact on hematopoietic differentiation. In a way that in a pre-leukemic setting, now, beside obviously using this as a kind of biomarker of something that is going bad, it would be very interesting to know how the different cell lineages are affected by this adaptive response, in a way that maybe lymphocytes can adapt to it better than other cells. So have you tried to look at just immunophenotyping, the different subsets of immune cells, to also learn more about how the different subsets respond to this adaptive genetics?
So the question, this is another point right now. We are doing this. We are performing in-depth immune phenotyping that actually we are doing in Vergata University with the Professor Voso in a different kind of setting of bone marrow failure syndromes, because right now we are aware of the fact that immune cells, T cell differentiation can play a pivotal role towards the inflammation. What we specifically found in SAMD9, it seems that we have a B-cell lymphopenia, NK deficiency, but also a switch, an imbalance between CD4 T cell and CD8. So it seems that we have a CD8 expansion in which we have more naive cells and maybe a defect towards the CD8 cell differentiation. And if we are thinking that SAMD9 and SAMD9L can have a pivotal role towards viral infection, so clearance of viral infection. And most of these patients used to start with a viral infection, not properly cleared, and then they establish the cytopenia. Maybe the immune defect can play a pivotal role towards the pathogenesis of the also hematological manifestations. So it's a crucial point.
Great. And just a curiosity, can you replicate all this in the mouse model? So do you see adaptive responses to germline mutations that are among the ones that you listed, or is it a very unique feature of humans?
We have SAMD9 and SAMD9L in mice. The problem is that being a gain-of-function mutation, it's really complex to replicate the model, because you cannot do a knockout mice, for instance. And it's also highly conservative gene. So that means that it's really difficult to predict which pathogenic mutations, which mutations are pathogenic and which they are not. This is the reason that most of them, even if you are following the ICMG classification, are variant of uncertain significance. And so trying to set a model, a mice model for this seems to be pretty hard. Okay. But this is what they are trying to do at Martin Brodarsky laboratory.
Okay, so thank you very much again, Enrico. And thank you also for being with us.
Thank you to you. Thanks.
Bye-bye.
Bye.
Okay, so we have the last speaker of the session, which is a newcomer, I understand. Welcome. So Ugo Chianese from University of Campania, Luigi Vanvitelli. So I guess you will just introduce yourself a bit on what you're doing.
Oh, sure. So, good afternoon. First of all, I would like to thank the organizers who gave me this floor, so the opportunity to share with you an overview of this project. I work as an assistant professor in Vanvitelli University in Naples. I work mostly with solid tumors, the characterization of solid tumors in the AP metabolic context. So just for give you an overview. About this project, yes, sorry. Yes, perfect. This is the laser point, I guess. Sorry. It's not working, this one. Okay.
Did you give him the presentation?
Yes, I sent. If you have any problems, I have also on USB pen drive. Yeah. But I sent you yesterday. Yeah? Okay, sorry. We solved this problem. Yeah, perfect. Still the remote is not working. Okay, it's working. Perfect. So, sorry for the inconvenience. As you know, pancreas is an amphicrine gland, and almost 90% of pancreatic tumors comes out from the exocrine part, specifically from the ductal cells. Critical issues related with this type of tumor are lack of symptoms that make the diagnosis very late with a high mortality rate. These aspects combined to an increasing incidence make PDAC a real medical urgency. Our aim is to investigate PDAC from a metabolic perspective. Generally, metabolism refers to the anabolic and catabolic reactions, so the production of large molecules and the ATP. Today, metabolism is a hallmark in cancer. However, tumors do not share the same metabolism, and several ambiguities make the scenario more complex. So we need to improve our knowledge on this tumor. For do that, we use single cell and spatial transcriptome sequencing. Then we move to in vitro validation to confirm our hypothesis. By starting from single-cell RNA-seq data from tumor samples and healthy donor, using a dual clustering approach, we were able to associate the metabolic landscape to the cellular subtypes. Then, focusing on the ductal cells, we observed an enrichment for normal cells in mitochondrial-related processes, instead of tumors that showed a predominance of glycolytic activity. And notably, the expression of the glycolytic pathway was showed interpatient heterogeneity in both terms of expression level and metabolic enrichment as well. By preserving the structure, architecture of a tumor with spatial transcriptomic, in addition observing an immunosuppressed microenvironment as a function of a disease stage, we were still able to detect glycolysis as involved in biological processes of tumor cells, and as observed at single cell level, the expression of the glycolytic signature was still heterogeneous between patients. So we investigate how the glycolytic expression profile is associated to the clinical outcome in a larger cohort from TCGA data. From TCGA data, by applying our glycolytic signature, we were still able to stratify patients based on the expression profile. And these reflected the glycolytic enrichment and positively correlated to the overall survival. Specifically, how much higher was the phenotype, how much was the prognosis. We also validated these into two independent cohorts, observing the same trend, and we were also able to identify the glycolytic phenotype as enriched for basal-like subtypes, that is the most aggressive one, and we correlated it with the hypoxia status. Lastly, thinking in a translational way, we moved from the bed to the bench side we were looking for some in vitro model that was able to recapitulate the heterogeneity observed in patients, something that we can control and monitoring. So we started by analyzing RNA-seq data from several PDAC cell lines. Applying the same signature, we were still able to observe heterogeneity. So thinking to interfere with the glycolytic activity, we use one of those genes as possible target that we identify in LADH-A as functional endpoint of glycolysis. So once that we confirm its expression at protein level as well, we assess the glycolytic activity and the proliferation upon treatment. From the glycolytic activity, we observed the significant results in the high systems, high glycolytic systems, while no significant effect was observed in the low one. Similarly, we had an anti-proliferative effect, most efficacy in the high systems. We also further investigate these via integration of multiomics data. From metabolomic analysis, we observed that the lactate, so the final product of glycolysis, was significantly reduced in the high system, while no significant effect was observed in the low one. Currently, from proteomic analysis, glycolysis and glycolytic-related processes such as hypoxia were significantly reduced, while no modulation was observed in the low one, meaning that the metabolic treatment significantly affect the energy and the proliferative capacity of the high glycolytic phenotypes that clinically are associated to the worst prognosis. So, taking on message, PDAC exhibits metabolic heterogeneity associated with tumor aggressiveness and sensitivity to energy blockage. This evidence highlights the glycolytic signature as a stratification criteria to identify eligible candidates for target metabolic therapy. I would like to thank all the authors that contributed to this study, recently accepted for publication in a very top journal, and in particular Professor Altucci and Benedetti, my mentor, and obviously thank to you for your attention.
So if there are really no burning questions, I was told that we really have to move on to the next session. So first of all, I would like to thank you all again for being here. I mean, all the scientists that contributed and the ones that will contribute soon. I think the community is live, is growing, is exciting to listen to all these nice projects. And we look forward to meeting you again next year, and congratulations to you all for your presentations. And a particular thank to Claudio, who actually really made it today and gave us such an enlightening lecture. Okay, so now I was told that we have to start the second part. So I don't know if somebody will get into the room